Seminar series, the goal of which is to promote networking and collaboration from different disciplines to shift the paradigm from seeking a cure to developing a strategy of prevention. The topic of this inaugural year is inflammation and chronic disease, and this is the final standalone seminar in the series. We'll have a wrap-up event as part of 1GW in September. Please contact me at leaframe@gwu.edu if you'd like to contribute to that event and the panel. Today, we are diving into treatment approaches with a presentation on metabolic control of inflammation and infection from Vishwadeep Dixit, DVM, PhD. Dr. Dixit is the Waldemar von Zettwitz Professor of Comparative Medicine and of Immunobiology at Yale School of Medicine. His research is focused on understanding the interactions between metabolic and immune systems with the goal to reveal molecular targets that can be harnessed to control inflammation and immune dysfunction as means to enhance the health span. The research in Dixit Laboratory is funded by the NIH, Glenn Foundation for Aging Research, and the Cure for Alzheimer's Foundation. We are thrilled to welcome Dr. Dixit all the way from India today. And just a quick note before we move on, please enter your questions in the chat for Dr. Dixit to answer at the end. So with that, I will turn it over to you.
Thank you. Thank you, Lee. Hope all of you can hear me all right. And thanks again for the invitation and the opportunity to present some of our work to all of you. And so, like Lee mentioned, good afternoon to you all. This is evening for me here. I decided to travel to India, so my background is a little bit not exactly telling you where I am. So where I am is, I think my okay. Where I am actually is right here. If you can see this on the map, a city called Hisar, which is also my birthplace, in fact. And people used to ask me, "Oh, where are you from?" I said, "Oh, I'm from a small town famous for nothing." And it turned out that a couple years ago, there was this paper that was published in Cell, and what they found is exactly about 5 miles from my home. They discovered the largest unexcavated site of the Indus Valley civilization, where they discovered a skeleton, and with that they could trace the genetics of the origin of the people in Asia and South Asia. But this civilization is about 5,000 before Christ, and its script is still not deciphered. And it is thought to be one of the first civilizations that invented the use of baked bricks. So that's my hometown for you guys. But then here we are discussing chronic diseases. And for those of you who are not familiar with the statistics, this is something that is sometimes often gets lost as we focus on individual disease-specific approach. And what gets lost is that, in fact, aging itself is one of the greatest risk factors for chronic disease. And we think that inflammation has something to do with it. So what you're looking at here are the incidence of chronic diseases. And you can notice that as we begin to get older, something is altered in the biology that causes the emergence of almost all of these chronic diseases that are listed here, ranging from Alzheimer's disease, chronic kidney disease, diabetes, cancer, heart disease, of course, are the major ones. But we also know that not everybody that gets old gets these diseases. So there has to be some sort of underlying aging associated alterations in the biology that predisposes us to these various diseases. But then one also have to have a very specific trigger agent for those diseases to actually manifest. So for example, gout, which is also a disease of aging, happens when you have accumulation of urate crystals. That would induce inflammation, and joint inflammation is a major issue, as all of you know, with gout. And dyslipidemia is a trigger agent during aging to cause cardiovascular disease. And we all know that amyloid beta accumulation is responsible for, or at least is involved in, Alzheimer's pathogenesis. So all these multiple diseases, chronic diseases, are associated with aging. And actually, one of the arguments in the field is that, if we have all these age-related diseases that emerge simultaneously, is there something that is intrinsic in the biology of aging itself that predisposes us to that? And and if one could understand the mechanism of that aging process, then perhaps we can delay the onset of all the diseases simultaneously. Because what's happening now is that as we treat one disease at a time, with age, you basically exchange one disease with the other. So if one was to cure Alzheimer's, the likelihood of one getting cardiovascular disease remains the same. So that's the kind of the general, discussions that are occurring in the field of aging biology. But inflammation as a cause of aging associated degenerative diseases is actually nothing new, and in fact was first proposed way back, almost 120 years back, by Ilya Mechnikov, who is credited with the discovery of phagocytes or macrophages. And in fact, and he laid, of course, the foundations of innate immunology, but he was also the first scientist who coined the term gerontology, and he was extremely interested in longevity and biology of aging. And he wrote this rather controversial book while he was in Pasteur Institute called Prolongation of Life Optimistic Studies, where basically he predicted that the phagocytes are going to be extremely critical for the degenerative diseases of aging. And he writes in his book, is a quote from his book up here, that the phagocytes resist the influence of invading poisons better than many of the other cells, and sometimes are stimulated by them, producing disease. So what are these poisons that he was talking about, and what does this stimulation actually mean? And in fact, what you're looking at here is an electron micrograph of a macrophage that is isolated from the thymus of an aged mouse. And this image is basically proving what Ilya Mechnikov had predicted 120 years ago. So what you're looking at is a macrophage or a phagocytes that has accumulated multiple poisons, or the new terminology for these so-called poisons is damage-associated or danger-associated molecular patterns. So here is presence of lipid in this macrophage. We don't quite know whether this has been phagocytosed from the extracellular milieu, or whether macrophages are de novo synthesizing this lipid. There are presence of protein aggregates within the cytoplasm of the macrophages. This is the nucleus. It's pretty misshapen. And there are some suggestions in cell biology that a misshapen nucleus like this is a sign of a cell that is undergoing senescence. And then you see all this electron-dense material in the lysosomes. These are all signs of a cell that is undergoing massive damage. And then what's also intriguing is that you can see these structures that are present inside these macrophages. These are actually, in fact, crystals that are accumulating in these cells as the process of aging continues. And in fact, these are the poisons that Ilya Mechnikov was talking about. And these macrophages obviously do an amazing task of clearing all these damage-associated molecular patterns, so that the homeostasis can be maintained or can be restored. But obviously, the process of aging is such that it just never ceases. And humans evolutionarily have not lived more than 40 years, just like 300 years ago. So what is happening is that these phagocytes are accumulating these damage-associated molecular patterns and are getting activated. Now we know a whole lot about molecular machinery that senses these poisons or senses these damage-associated molecular patterns. And these are pattern recognition receptors, a concept that was first put forth by Charlie Janeway in our department at Yale. But in regards to aging, what's really fascinating is that all these diverse molecular patterns that I showed you, or these danger signals that I showed you, are sensed by a central cytosolic sensor, innate sensor, called as the NLRP3 inflammasome. And aging is associated with accumulation of multiple damage associated molecular patterns. Several of those are listed up here. These includes extracellular ATP, which is released when a cell dies necrotically, amyloid beta, certain lipids, lipotoxic lipids like free cholesterol, ceramides. These are all sensed through a canonical NLRP3 inflammasome dependent pathway. And what this does is that once the NLRP3 inflammasome is assembled by these damage-associated molecular patterns, it causes the activation of enzyme caspase-1, which is typically kept in a cell in an inactive state. And once caspase-1 is activated, it has multiple substrates, but two of its major substrates are pro-inflammatory cytokines, interleukin-1 beta and interleukin-18, which are kept in a cell, with, especially in macrophages, in a pro form, which is inactive form. And caspase-1 cleaves the IL-1 beta and IL-18, releasing IL-1 beta and IL-18 from the cell. And with aging, these two cytokines cause inflammation, and this process is called as inflammaging, a term that was coined by Claudio Franceschi several years ago. So what we and others have now shown that with age, macrophages, as they accumulate these poisons and continue to accumulate these poisons, they keep on tickling NLRP3 inflammasome in a chronic fashion, leading to a sustained low-level activation of caspase-1 via the NLRP3 pathway. And if one goes upstream and ablates NLRP3 and its adapter, ASC, then one can actually not only reduce the process of inflammation, but also downstream processes that we think are important for degenerative diseases of aging. So the animals that have reduced activation of NLRP3 inflammasome are protected from age-related loss of bone mass. They're also protected from glucose intolerance or development of type 2 diabetes. They have less adipose tissue inflammation. Their thymus remains more active and produces T cells till late in life. These animals also have reduced CNS inflammation. They're protected from age-related cognitive decline and also are less frail. So you would wonder, how come targeting just one innate sensor would have such a diverse response with age? And I think that really comes from the age-old prediction by Mechnikov that the phagocytes are really the critical cells that are responsible for age-related inflammation and diseases. And what happens is that each of these organs have resident macrophage cells that via the NLRP3 inflammasome get activated and produce disease. And so one of the organs that we have become extremely interested with age is the visceral adipose tissue. So what you're looking at is a CT scan that was shared by my colleague Darcy Johansson, who is now at the NIDDK. You're looking at a 31-year-old individual on the left and a 74-year-old individual at the right. Both these people have the same body mass index, but you can clearly notice that despite maintaining a healthy BMI of around 27.1, 27.9, the elderly have a much more increase in the visceral fat mass. And this happens in the process of normal aging. And what we know now is that this increased visceral adiposity with age in both males as well as females is linked or is at least associated with emergence of many diseases apart from just type 2 diabetes. It's also highly linked to cardiovascular disease. And we think that potentially visceral adipose tissue may be an important site where inflammation originates, and if we could understand what's actually going on in this expanded visceral adipose tissue of elderly, perhaps we can understand the mechanisms that produce inflammation. And one of the striking features of adipose tissue is that, in fact, adipose tissue is not just a tissue that stores energy in form of triglycerides and produces hormones like leptin and adiponectin and adipsin and several other adipokines, But in fact, adipose tissue is also an immunological organ. Okay, so what you're looking at here is a healthy mouse, a young healthy mouse, where everything in green that you're seeing is a myeloid cell that is indelibly marked with GFP. And so you can see that in healthy animals, the adipose tissue, and this is a whole mount confocal image, that the healthy adipose tissue is actually loaded with myeloid cells, and majority of these are in fact macrophages. So what we think is that in fact these are normal components of the adipose tissue, these macrophages, and play a critical role in maintenance of adipose tissue function, in maintenance of homeostasis, and perhaps with age, these cells, the activation status of these cells, perhaps via NLRP3 inflammasome, gets altered, and then may produce disease. But important thing to note here is that in fact the macrophages or immune cells that are present in the adipose tissue are not just randomly distributed. In fact, they are present in very unique niches. And as the field is now maturing, we are beginning to realize that the presence of these cells in these niches is absolutely critical to how these cells interact in terms of regulating organ function, not only organ function, in fact, whole body, and also influence organismal metabolism. So what you're looking at in green are these macrophages that are hugging these structures, which are in fact nerve fibers. So these are sympathetic nerves, and macrophages are also present in these niches. As you can see, they're tightly hugging these nerves, and this is a live video 2-photon microscopy of a healthy mouse, where green cells are myeloid cells. In red are, in this case, are the nerve fibers, and then there are blood vessels here. And you can see that this in a 2-hour time-lapse video, there are certain macrophages that are dedicated to live on the nerves. You can see these fellows right over here, and they are putting their dendrites or processes and surveilling these nerves. But the interesting thing is that these cells are actually not very motile. They're not going up and down the nerves. They're actually sticking to where they are, and are doing something that is actually very intriguing. So we discovered these cells together with a group in Portugal in 2017, and we call them nerve-associated macrophages. And what these macrophages do is actually a very interesting and a very important function in terms of regulating the neural control of the adipose tissue by maintaining the amount of catecholamines or the neurotransmitters that are available for the adipose tissue. So what happens is that the macrophages uptake the neurotransmitters like norepinephrine, and that with age, what happens that the NLRP3 inflammasome is activated, and this NLRP3 inflammasome upregulates enzymatic machinery, including this enzyme called as monoamine oxidase A in the macrophages, which is critical for the breakdown of norepinephrine. So basically, with age, macrophages suck up this norepinephrine that is produced from the sympathetic nerves, and but through increase of this MAOA, they basically degrade it. So that when your nerves produce these catecholamines, so that these catecholamines can activate the process of lipolysis. In fact, these inflamed macrophages at this point of time take these neurotransmitters and break them down, so that in fact adipocytes don't see it. So your neurotransmitters don't work very well, and that's one of the mechanisms whereby elderly are much more prone to metabolic dysfunction and are unable to utilize the resources like triglycerides in the adipose tissue. Then another important feature of inflammation with age is accumulation of B cells that actually also happen within the adipose tissue in these unique niches. And this niche here, for example, is a tertiary lymphoid follicle, which forms with age. It's called as fat-associated lymphoid cluster. And what you're looking at in yellow are B cells. And we've shown that with age, The NLRP3 inflammasome, via the release of IL-1 beta and IL-18, expands these B cells, which are aged B cells. These are very unique cells. I don't have time to go into the details of that. But if you can block this process of accumulation of these aged B cells, you can actually restore the insulin sensitivity and ability of the aged adipose tissue to undergo lipolysis. So in fact, the immune system and the immune cells that are present within the adipose tissue play a very critical role, in not only in just control of inflammation, but actually maintenance of organismal metabolism. So what we decided to do was to really kind of look into it in a deeper fashion and characterize what is actually going on in the adipose tissue when it comes to the resident immune system. And this work was done by my postdoctoral fellow in my lab, Emily Kohlberg, who has just now moved to UCSF to start her own lab. So what Emily decided to do was to look into the tissue resident immune system. And one can do that by intravenously injecting an antibody, which is labeled with FITC, intravenously into a mouse. And so that this antibody labels everything that is present, all the immune cells that are present in the blood. And then you can basically exclude those green cells and sort the cells that are only present in the tissue parenchyma, thereby looking only into the tissue resident cells and not the contaminating blood cells. By the way, adipose tissue is highly vascularized organ. So if you just digest and do a flow cytometry of adipose tissue, you will get a mixture of cells that are present circulating through in the adipose tissue versus the cells that are in the tissue parenchyma. So, and we conducted single cell RNA sequencing analysis of the tissue resident cells. You're looking at, on the left, are young animals where we have isolated the tissue resident leukocytes from the visceral fat, and in the old, and on the right are the old. So, I mean, it is pretty striking. I think everybody in the audience can see it fairly clearly that there are massive changes that occur as the process of aging goes on. And some of these changes, in fact, are almost binary, and we were pretty excited to see those. So, for example, some cells expand and the other cells contract with aging. The cells that expand with age are the T and B cells. So these are alpha beta T cells right here, cluster 1, and the B cells here, 10 and 11, they expand as well. Macrophages, which in fact are supposed to, increase, which increase, for example, in obesity in the adipose tissue, it's not the same in aging. And sometimes people get confused that obesity is accelerated aging. In fact, that is not the case when it comes to immune system. So in fact, macrophages with age in adipose tissue, unlike obesity, don't expand. In fact, they decline. We lose these tissue resident macrophages. But the cell that caught our attention quite a bit is these innate lymphocytes, or ILC2s. They almost disappear with age. And this is just a summary of the characterization of the tissue resident compartment with age. So what you get is expansion of effector T cells, as well as tissue regulatory cells, Tregs. And there is increase in the pro-inflammatory B cells, which are present in the fat-associated lymphoid clusters. And then there are a bunch of cells that are lost with age. This includes gamma delta T cells, their loss of NK and NKT subsets, as well as ILC2s. And then there is a loss of homeostatic macrophages with age. Given the fact that we saw a dramatic loss of ILC2s, so we decided to follow up on these cells, and I'll show you the data on these cells as to what that actually means for aging. So this is just the orthogonal validation of the single cell results that I just showed you, that in fact, if you do the flow cytometry of young and old, you can clearly see that the ILC2s decline. They decline not just in one sex, they're declining both in males as well as females. And this is actually a very important point, which is often ignored, that male and female immune system is not the same. There are major differences in how these immune cells get altered with tissue aging. For those of you who are not very familiar with ILC2s, These are innate lymphocytes, and they are similar to Th2-like cells, Th2-like lymphocytes. And the key feature of these ILC2s is that they can influence multiple cell types through production of IL-5, for example. They can expand the eosinophils and influence the function of macrophages and mast cells through IL-13, including dendritic cells, can act on B cells via IL-6, and also activate T cells through MHC IL-13 complexes. And these cells got quite a bit of attention a few years ago when a few labs reported that, in fact, these ILC2 cells are pretty important in terms of increasing the energy expenditure or uncoupling the mitochondria in the adipose tissue. So there is this hypothesis in the field that if you convert these white adipocytes into brown adipocytes, which are adipocytes that have high expression of mitochondria containing UCP1 protein, uncoupling protein, that instead of producing heat would produce, instead of producing at ATP will produce heat as a means to kind of cause weight loss. So basically that's what one thinks is important. And then there is the cytokine called as IL-33, which acts on the ILC2s. It's produced from the stromal cells. It acts on ILC2s, expands the ILC2s, and then ILC2s are supposed to produce IL-5 to act on eosinophils, which then through STAT4 dependent mechanism converts a white adipocyte into a beige-like adipocyte, which is a thermogenic adipocyte. And it's shown here, for example, that if you inject animals with IL-33 in red, you see increase in energy expenditure. This is oxygen saturation, relative oxygen in the metabolic chambers. So obviously, it would suggest that, perhaps we are losing the ILC2s, and the loss of ILC2s in aging could thus be very critical in perhaps causing the metabolic dysfunction and promoting inflammation with age. So the obvious experiment was to actually treat the old animals with IL-33 and see if we can actually restore the loss of ILC2s. And this was very promising. In fact, we can do that pretty efficiently. So again, you're looking at ILC2s with age. These are 24-month-old animals, which is analogous to a roughly 75-year-old human. These cells decline pretty dramatically. And if you give these animals IL-33 injection, and you look at 2 days post-injection, you can see that the ILC2s are back up to the young levels. And these levels of ILC2s actually remain high even 6 weeks post single IL-33 injection. So it's pretty encouraging. So we thought that, oh well, that's the way to perhaps cure aging, and we were pretty excited about that. But I'll show you the data, which is not that that clear in terms of curing anything, in my view. So, which is that, so here, when we treat these old animals with IL-33 and look at what is actually happening in the adipose tissue and the whole body metabolism with energy expenditure and thermogenesis, in fact, there is absolutely no difference in the animals that are treated with the sham versus IL-33, where we see the expansion of ILC2 cells. Okay, and so we thought that what is actually going on? We are getting the ILC2 expansions, but IL-33 is not, increasing energy expenditure. So what is going on with IL-33 and its receptor? So the obvious explanation would be, IL-33 levels decline with age, and some people have actually reported that in different tissues. But when we actually look at IL-33, in fact, IL-33 levels increase with age. But what also happens is that the soluble IL-33 receptor increases pretty dramatically with age. So what we're thinking is that perhaps the IL-33 bioavailability in the adipose tissue is impaired as the process of aging continues. And so we were thinking that perhaps there could also be a signaling issue, that maybe there is so-called IL-33 resistance, that IL-33 is not signaling. So what we decided to do was just treat adult animals with IL-33 and look at the signaling here, in this case phosphostat 3. And and treated the old animals with IL-33 and looked at phospho-STAT3, and you can see that there's actually equivalent activation of phospho-STAT3 when we provide exogenous IL-33. So there is really no signaling deficiency when it comes to age. But what is actually happening is that when we look at the stromal cells of young and old animals, the IL-33 in young is produced from these mesenchymal cells that are PDGF receptor alpha positive. Okay, but with age, what is happening is that this population of mesenchymal cells that are supposed to produce IL-33 no longer produce IL-33. In fact, the IL-33 now is produced from another stromal subset of mesothelial cells. So aging switches the source of IL-33 from mesenchymal to mesothelial, and we think this has something to do with the lack of expansion of ILC2s that happens over the course of aging. So the obvious experiment was to see what would happen if, because previous studies have shown that if you give IL-33 to young animals, you increase thermogenesis, or basically uncouple the mitochondria, and those animals are protected from cold challenge, because then these animals given IL-33 can increase their core body temperature. Okay, so here are old animals that are young. So let me just lead you through this. In these triangles are adults that are given PBS, and blue line with these red triangles are adult IL-33 treated animals. And old animals are the ones shown here. And what was pretty striking was that, in fact, with age, what we know is that IL-33 treated old animals, in fact, not only were not protected from cold shock, but in fact died much more rapidly after IL-33. And in fact, IL-33 injections in old animals, despite expanding the ILC2s, do not cause any change in the uncoupling protein. Okay, so we decided to sort these ILC2s out post IL-33 treatment to see what has IL-33 now done to these ILC2s, that when we expand these ILC2s, that we are actually in fact killing the old mice instead of protecting them. And it turned out that the ILC2s that expand with age after IL-33, in fact, are pathogenic. What happens is that, we have pathogenic cells that remain, and when we give IL-33, we basically expand more of those bad guys. And this is apparent from this RNA sequencing analysis. And the major pathways that get upregulated are pathways that are involved in the increase in the pro-inflammatory cytokines, including IL-6 family cytokines. And then there are pathways that get downregulated that are important for maintenance of homeostasis in the ILC2s. Okay, so so what we decided to do was to see whether the young ILC2s are protective, whether they are the ones that can, if we provide those or adoptively transfer those young ILC2s into old animals, what impact would that have? So we in fact did that. We expanded the ILC2s in young animals, sorted them out, gave them to the old animals. So as I showed you before, the old animals die much more rapidly when it compared to the young animals when you challenge them with cold shock. But these old animals, if you transfer the young ILC2s, these animals are now protected. So in fact, ILC2s are indeed protective, but they are protective only if they're provided from a young microenvironment, and not the expanded ILC2s from the aged animals. So what I've shown you is that visceral adipose tissue is actually an immunological organ. It's a very major immunological organ. In fact, every lineage of hematopoietic system is present within the fat. And that visceral adipose tissue, through activation of these resident immune cells, is a major source of inflammation in aged animals and perhaps in humans. What I also showed you is that aging causes a loss of protective ILC2 cells. Whatever ILC2s remain in aging, if you expand them through IL-33, in fact, that just expands the pathogenic ILC2s and impair thermogenesis, and actually is detrimental to the health of the animal. Again, proving that the resident immune system and the metabolic system play a very critical role in maintenance of homeostasis. So this obviously raises the question as to what could be the processes that we can understand with regard to tackling age-related disease. And one of the things which is pretty well established since several years is the longevity that happens when the host is in negative energy balance, which basically has been shown in the context of calorie restriction. So if the animals are calorie restricted, they live longer, they maintain homeostasis, they have less diseases, but that also comes with these trade-offs. These trade-offs are there's perhaps loss of immunological regulation. They're supposed to be more prone to infections. There's also reduction in reproduction and growth. But so what we wanted to see was when a host undergoes negative energy balance, and it has to have an integrated immunometabolic response where when the glucose is less, it has to burn now fatty acids. And we want to see what happens in this context when the host has to now utilize alternative metabolic fuels like fatty acids and ketone bodies. Can we harness these alternative metabolic fuels in terms of regulating the process of aging and targeting inflammation? So again, I think I don't have to go into the details because in your program, you're well aware of the process of ketogenesis, which is absolutely essential for mammalian survival because beta hydroxybutyrate, one of the major ketone bodies, serve as an alternative fuel source when glucose is less. And glucose is less when you're starving. And as you know, mammals have probably starved through multiple phases during their life. And during those times, BHB or beta hydroxybutyrate serves as the alternate fuel. It enters the TCA and drives the electron transport chain and release of ATP. So we decided that we're going to induce a state of ketogenesis in an animal to kind of mimic a state of starvation without actually causing starvation. So animals were fed a diet which is rich in medium chain triglycerides and low in carbohydrates. And we decided to do the single cell analysis to see what we have done now to the resident immune system. So as you recall that ILCs are lost with age, but when we give these animals ketogenic diet, the cells that we expand are the ILCs, including the gamma delta T cells that are shown here. And I'll show you some data with regard to gamma delta T cells now with ketogenesis. And we have previously shown that in fact ketone bodies are pretty important triggers for deactivating the inflammasome or the NLRP3 inflammasome. So what you're looking at is the inflammasome activation assay. These are macrophages that have been activated via the NLRP3 inflammasome with LPS and extracellular ATP, and in presence of increasing concentrations of beta hydroxybutyrate. 4 millimolar of beta hydroxybutyrate is something that you would get when you're in a state of fasting. Between 5 and 10 is when you reach a state of starvation. But you can see that the physiological ketogenesis in animals can pretty effectively downregulate the activation of the NLRP3 inflammasome, which is shown in this case with ATP. And it also happens when you activate the inflammasome by urate crystals. So the way the beta hydroxybutyrate works is that so when a host is in this starvation state, or you could induce that by a low carb diet, when you don't have enough glucose, then now host, instead of using glycolysis, has to use fatty acid oxidation. And these fatty acids are converted into ketone bodies because these long chain fatty acids cannot cross the blood-brain barrier. Okay, so you have to produce this beta hydroxybutyrate, which has to now serve essential, maintain survival of the host. And the two organs that really require BHB at that point is brain and the heart, which are major consumers of ATP. But BHB also then act on the immune system. And we have shown that the way BHB reduces the inflammation, and especially is through the inhibition of the NLRP3 inflammasome, that it actually does so in a very simple fashion, that it serves as a kind of a small molecule which prevents the oligomerization of the adapter and preventing the efflux of the potassium in response to all these dams that are shown here. Thus does not allow the assembly of the NLRP3 inflammasome to occur and preventing inflammation. So the obvious question from this work that emerged was whether ketogenic switch could be employed to regulate inflammation during the process of aging. And if indeed that is the case, would that protect against infection, or would that actually increase the risk of infection? Because we know that inflammation has a very important role, not only in terms of maintaining homeostasis, but also for mounting a proper response to infections. And the model that we employed to study this, and now this is exactly a year when WHO called COVID-19 a global pandemic, March 11th. And so we decided to study, create a model of aging where we could study the COVID-19 like pathology. And the model that we chose is this natural mouse beta coronavirus called as MHV-A59. A virus which is in the same clade as the SARS-CoV-2, is very similar to SARS-CoV-2 virus, except that MHV-A59 do not utilize ACE2 as the entry receptor. So which means that we can actually infect regular black 6 animals with MHV-A59 and study the pathology that it may cause. So here we can see that in fact MHV-A59 enters the lungs, and you can see the virions in the lungs of these old animals. And very similar to what happened in people, where elderly are much more prone to death from SARS-CoV-2, that the old animals that are infected with mouse beta coronavirus in here in red, with equivalent dose of infection dose, succumb to infection pretty rapidly, whereas the young animals are able to survive. And what we noticed in this scenario is that in the lungs, the cells that are lost, the majority of the cells that are lost are in fact the gamma delta cells. So these are young animals that are infected with MHV, and these are old animals that are infected with MHV. So you can see that the old MHV animals have much fewer gamma delta T cells. And I'll get to that in terms of what that could potentially mean for aging and protection from infection. But first, to kind of really prove to you that this is a model that is quite relevant for aging, is that these old animals, old black 6 mice that are infected, lose much more body weight. So they have similar sickness behavior that occurs in humans. They don't eat much. There's anorexia, there's weight loss. This is the oxygen saturation. You can see that the animals have much greater loss of oxygen saturation. Old animals have much greater loss of oxygen saturation. And similar to what is seen in elderly, there's increase in lung inflammation. This phenomena called as ARDS, acute respiratory distress syndrome, where there is accumulation of mucus as well as infiltrating immune cells shown in these arrowheads in the lungs of these animals. Just to further show that similar to elderly, where there is increase in cytokine storm, the lungs of these old animals have more of these pro-inflammatory cytokines. They also have increase in the inflammasome activation. So, and the marker of the inflammasome activation is the activation of the P20 enzymatically active caspase-1 heterodimer. So you can see that old infected animals have more of this. And, and this obviously led us into the question that I posed before, is whether if we engage a ketogenic switch in these animals, would it be possible to affect the disease outcome in aged aged animals. So these are old animals, 24-month-old animals given a chow diet and a ketogenic diet for a period of about 7 days. And you can see that the old animals that are given a chow diet rapidly lose body weight. About 15% of the body weight is lost by seven days. But the old animals given the ketogenic diet are protected from it. Weight loss and anorexia. These old animals are also protected from loss of oxygen saturation. And what was pretty exciting was that, in fact, in the lungs of these old animals, ketogenic diet was able to restore the gamma delta T cells that were lost post-infection. Given our previous work and the fact that inflammasome is increased or inflammasome is activated in the coronavirus infection, we wanted to see whether ketones itself, in response to the beta coronavirus infection, can block the inflammasome activation. And in fact, BHB does so very efficiently. What you're looking at is the inflammasome activation assembly assay, where we're looking at the dimerization of the adapter for this inflammasome ASC. In this case, these macrophages are infected with A59 virus and given the BHB. And you can see that it reduces the monomers as well as the dimers of the ASC complexes. And indeed, when you look in vivo, the ketogenic diet-fed animals post-infections have a significant reduction in the inflammasome activation as measured by the active caspase-1. And the age-related increase in cytokine storm that occurs in multiple organs is also reduced in animals given a ketogenic diet. In this case, in blue, these are ketogenic diet-fed animals. You're looking at lung, visceral adipose tissue, and hypothalamus. This is IL-1 beta. This is the NLRP3, and you can see a dramatic reduction in these pro-inflammatory cytokines. And then to kind of really see and understand the mechanism as to how ketogenic diet is protecting against this coronavirus infection in animals, we conducted the single cell sequencing in the lung. These are lung homogenates of animals that are ketogenic. And I think it's pretty dramatic, the effects that we observed. And one of the key cells that is lost or is prevented from expanding in aged animals post-ketogenesis is these pro-inflammatory neutrophils. And these pro-inflammatory neutrophils are also cells that are very pathogenic in human lungs. And the other cell type that is prevented from expansion are these pro-inflammatory monocytes. So so ketogenesis prevents these two innate immune myeloid cells from expanding and causing damage in the lung. But what was also very clear is that indeed, as we had seen before with flow cytometry, that the single cell analysis also revealed that ketogenesis expands the tissue protective gamma delta T cells. These are in the infected animals. And that in fact, these gamma delta cells have an expansion of these mitochondrial pathways that are important in electron transport. And you must be wondering why gamma delta T cells should be important here in COVID. And now I think we have enough data out there, including these data from 2 different cohorts, that that the gamma delta cells in the blood are lost in patients with COVID, and this is directly associated with the severity of the disease. So I will end by just showing you the importance of gamma delta T cells in controlling the infection. So we have previously shown that in fact, ketogenesis or ketogenic diet can not only prevent infection of coronavirus, but can also protect against flu infection. So in black are flu-infected animals, and in red are flu-infected animals that are given a ketogenic diet. And you can see that these animals are protected from weight loss as well as mortality. And by day 4, almost all the, not almost, all of the chow-fed animals are dead, whereas about 50% of the ketogenic diet mice survive. And during flu infection, also ketogenic diet expands the gamma delta T cells. And if you knock out the gamma delta T cells and give the mice ketogenic diet and then infect the animals, okay? So these are, for example, if gamma delta cells are absent, what you get is increased mortality. So for ketogenic diet to protect against flu infection, at least we know against flu infection, it requires the presence of gamma delta cells. Okay, so what I've shown you is that, in fact, that ketogenesis is indeed a very important dietary switch or a metabolic switch that can impact the immune system. So, for example, in this case, I've given you the example of COVID as well as flu. So, with compared to young animals, when young animals are infected, there is an increase of these pro-inflammatory cytokines, but these cytokines then decline, so that they do not remain sustainedly high in the young animals, and this causes control. So these cytokines then control the infection by protecting the host by expanding the gamma delta T cells and expanding the protective monocytes. But with age, what is happening is that there is a chronic inflammasome mediated increase in pro-inflammatory cytokines, and which is responsible for causing severe COVID in aged. And if we change the substrates that are available for these cells, for example, by switching the diet from more of a glucose to a beta hydroxybutyrate source, what you get is a protection from COVID and protection from pro-inflammatory damage that occurs in these animals. And this happens in part via expansion of the gamma delta T cells. So I will end here and thank you all for your attention, and most importantly, thank my colleagues from my lab and everybody, my collaborators that have helped get this work done. So as I showed you, the initial part of the work with aging and inflammasome was done actually by multiple people in my group. Initial work was done by Uni Yom in my lab and several past members, Christina Kamel, who is now at University of Minnesota running her own lab, and Ryan Grant, and who was in Purdue. And the rest of the work that I showed you with the ILC2s was done by Emily Goldberg, who I mentioned now is at UCSF and is pursuing her interest in relation to immunometabolism and aging. And the last half of the work with the establishment of the COVID-19 model on mice was done by postdoctoral fellow Sunjin Roo, who very diligently worked to establish this model and demonstrate the effects of role of ketogenesis in aging. The COVID model was developed in collaboration with my colleague and friend Andrew Wang. Akiko Iwasaki in our department was critical in providing us help and advice in relation to flu experiments. And I also need to thank Max Ardemov, who helped us with the computational analysis, and the funding agencies and collaborators that are on this slide. So thank you again for your attention. I wish I was there in person with you, but I'll be happy to answer any questions in a few remaining minutes.
Yes, thank you. That was really a wonderful talk. And we do have a few questions lined up for you already. The first one is a two-part, so I'll give you the first part first. What about non-visceral fat in regards to immune response?
Yeah, so one of the things that happen with age is, in fact, the visceral adipose tissue expands, but the sub-Q adipose tissue, which is supposed to be more protective, in fact, declines. And the majority of the inflammatory changes that occur with age occur in the visceral adipose tissue, not so much in terms of inflammation in the sub-Q fat. So, for example, these populations that I showed you, of course, there are changes in the sub-Q fat, but the resident immune system changes in the visceral fat are much more prominent and pro-inflammatory.
That makes a lot of sense. And I love this question. Is there a critical link to the gut microbiome?
Yes, I get this question a lot. I'm pretty sure there is an important role for gut microbiome and alterations in the microbiota that could play a role in emergence of inflammation that occur not only in multiple diseases, but including aging. because there has been this hypothesis that there is a leaky gut which happens with age. And if you give these dyes to these old flies, they become smurfy, which means they basically become blue. However, I think we should be a bit cautious when we interpret those data, because to date, there is really no evidence that humans have leaky guts. Elderly humans have leaky guts. Yes, mice may have that, flies may have that, but the evidence in terms of humans is still lacking, or is not there yet. But we do also see, for example, that there is a possibility that the gut microbiota could potentially, or the pathogen associated molecular patterns from the gut microbiota could leak into, via the portal circulation, into the visceral adipose tissue, and could cause the formation of fat associated lymphoid clusters. We do not have definitive evidence at this point of time to say how much is the contribution of the gut microbiome. But perhaps if you call me in a year or two, I can come to GW and show you the data as to what exactly is the contribution of microbiota. But I do think that it plays an important role, but we have to study it very carefully.
Excellent. And we're going to take you up on that offer for sure. We would love to hear that talk. Another question, does ILC2 restoration expansion via IL-33 impact mouse feeding, body weight over time?
Yeah, so that's actually a very important question. There's really no effect on any of these parameters. So the animals are not sick, they're moving fine, they're drinking okay, they're consuming the same amount of food, except that they're not thermogenic. They expand ILC2s, and when those ILC2s expand, it kills the old animals. So don't give IL-33 to old animals or old humans.
Follow-up question, can you please comment on your experimental aging time course and distinguishing between normal aging related to cellular senescence versus effects of inflammasome-mediated mechanisms?
Yeah, so I think that's an important point. So the normal aging process itself is, in terms of inflammation that ensues in a mouse which lives around about 3 years, you begin to see the inflammasome activation. We've actually published that in our prior papers. You're happy to look it up. It says, you begin to see the increase in IL-1 beta and increase in pro-inflammatory cytokines and inflammasome activation roughly around 12 to 14 months of age in a mouse. And from that point onwards, it remains high and keeps on going. And that's the course of normal aging process, because I mean, if you really see that eventually, as the aging process is basically accumulation of damage, cells just keep on accumulating damage as just their, I guess, cost of working, and and the macrophages eventually get overwhelmed in clearing up these cells. The macrophages are not there to kill us. Inflammasome obviously is not there to kill us. And it is that these mechanisms eventually get overwhelmed, these protective mechanisms get overwhelmed when there remains sustained activation and cause then trade-offs in terms of tissue protection. So that is normal aging process. Now you could superimpose that with a disease and give normally aged animals or aging animals a specific trigger agent, which could be urate crystals, which could be amyloid, and then you can actually accelerate or induce a very specific disease. And we and others have done that in the context of aging.
We have one last question. How is IL-33 regulated? Any known upstream regulators?
Yeah, that's a good question. I think in context of aging, we've just begun this work, and I think at this point of time, I really do not have a very clear answer as to what actually regulates IL-33, because if you see, IL-33 levels are in fact higher with age. In the visceral adipose tissue, unlike what would think that it declines, it does not actually decline. But what is happening is the source of IL-33 is changing. The cells that are supposed to produce IL-33 no longer produce it, but other cells produce it. But what that really means is that has the IL-33 itself changed? or if the change of the source has changed, it means the cells that are present in that niche are no longer seeing what they're supposed to see. So I think there are too many questions at this point of time in regards to this very interesting process that is occurring, and I think the time will tell as to the details of how IL-33 mediated homeostasis in aging and ILC2s is perturbed.
Well, I think that's the perfect place to end. There's more research to come. Thank you so much for joining us. We really appreciate it, and it was a wonderful talk. And thank you all for joining us as well.
All right. Thank you, everyone. Thanks very much again. Bye-bye.
Bye, everyone.